video, I'm going to be talking again about SCARA5 and SCARA5 and how it prevents a cascade of iron-driven redox and adhesion pathology by forcing iron to remain ferritin buffered instead of entering the cells. And I'll be talking about some of the mechanisms, but I'll try to keep it basic and not too technical. Now, some nuts and seeds, they can be high in PUFAs, or polyunsaturated fatty acids, and these can lead to lipid peroxidation. But SCARA5, it can prevent iron or Fe2-driven generation of lipid radicals in polyunsaturated fatty acid-rich membranes, and this ends up protecting plasma membrane lipid rafts, protects lysosomal membranes, and also protects endosomal membranes where integrins recycle. Now, I'll be touching on the lysosomes. If you've seen my other videos on lysosomal membrane peroxidation, you already know that if it is peroxidized, then the cathepsins leak out, and then the lysosomes cannot degrade the exosomes because the cathepsins are supposed to degrade the exosomes inside the lysosome. But the cathepsins have to stay in the lysosome in order for that to be possible. Because if the exosomes are not degraded, then they will go to the tumors, and that is terrifyingly effective. Now, a SCARA5, if it's intact, it also protects against weak adhesion in the immune cells, or prevents immune cell dysregulation, because SCARA5 also affects integrins, nanoclustering, and cytoskeleton, so prevents the oxidation of integrin, beta-1, beta-3 misfolding, and talon-kindlin dissociation, maintaining actin-integrin coupling. Now, it's critical for integrins beta-1, beta-3 to stay properly folded and clustered, and these integrins, they maintain correct conformation as well as nanoscale clustering, essential for strong adhesion and signaling. Also, talon-kindlin remain bound to integrins and sustains integrin activation, which is called inside-out signaling, and supports force transmission, which is a part of immune synapses. And it's critical to enable the cytoskeletal tension as a result of the actin-integrin coupling being preserved, because it is critical for PMAC formation, which is short for peripheral supramolecular activation cluster in immune synapses. And you already know what that means. If you've seen my other videos on PMAC formation, I've made other ones. Tumors, they hate PMAC because it is the end of the tumor. So anything that supports SCARA5 also, or SCARA5, will also support PMAC formation. So SCARA5, allowing PMAC formation, will allow a stable adhesion between T cell or NK cell and tumor cell, because an immune synapse is basically a connection, or like an anchoring point between a T cell or NK cell and a tumor cell. So fibronectin fibrogenesis is maintained, and cells can remodel the extracellular matrix. If you've seen my other videos, you already know what that is, ECM for short, and then also supports migration, anchoring, and immune infiltration into tumors. And without SCARA5, loss of beta-1, beta-3 integrin, leading to misfolding, so therefore PMAC doesn't work. Talon-kindlin dissociation, which weakens integrin cytoplasmic tail interactions, therefore impaired linkage to actin. Therefore, no PMAC, because actin or actin polymerization is another major factor of PMAC formation. So no SCARA5 means no actin. Okay, now on to mitochondria and lysosomes. SCARA5 prevents mitochondrial and lysosomal rupture. Therefore, ATP is available for immune cells, and the lysosomes can degrade exosomes so that tumors do not get the exosomes. And the way SCARA5 protects mitochondria and lysosome is because SCARA5 keeps iron sequestered in ferritin. So that way, the iron does not cause an accumulation of reactive oxygen species and doesn't cause damage to the mitochondria and does not leak out of the lysosome. Or in other words, SCARA5 prevents iron overload entering lysosomes in the first place, because if iron did do so or overloaded lysosomes, the lysosome would become peroxidized or permeabilization enabled, therefore cathepsin leakage, and then exosomes cannot be degraded by the cathepsins if they leak out. Okay, I'm going to name off some beans here um, from top to bottom that have the strongest support for SCARA5. So the first bean here is black beans, very high when it comes to supportive strength for SCARA5. Bean number 2 is small red beans, also high for SCARA5 support strength. Third bean here is Anasazi beans, not as high as black beans or small red beans, but pretty moderate, so they're pretty high. Fourth beans is red kidney beans, up there with Anasazi as well. And then the next beans are not as strong when it comes to supporting SCARA5, but they are moderate, and that is pinto beans. So that sits at number 5. Sixth bean here is pink beans, moderate. And then the weakest beans here for SCARA5 support, which means that these following beans are not going to be that great to support the PMAC formation, which tumors hate. And those weak beans are cannellini beans, a low to moderate, white kidney beans, a low to moderate. Great northern beans are pretty low when it comes to SCARA5 support, but great northern beans are great for the lysosomes so that the cholesterol is not being loaded and full, or so that the cholesterol is not loading up the lysosomes and packed full of cholesterol into the lysosomes, which great northern beans prevents. Tumors are also known for packing or loading up cholesterol into the lysosomes to make the lysosomes dysfunctional. Therefore, the lysosomes cannot degrade the exosomes, and then the tumors receive the exosomes. But great northern beans can help prevent that. And the weakest beans here, when it comes to SCARA5 support, would be navy beans. So that's it for the video. If you want more future videos, then make sure to like, comment, share, subscribe. Keep it locked in here on my channel, and I'll see you in the next video.